Hello viewers, I am Dr. Robiul. I work as a lecturer in pathology in a medical college hospital, and I am making this video for my students and also for you. Hope someone finds this helpful. Today's topic is angiogenesis. This video will contain the definition of angiogenesis, followed by some common terms, different roles of angiogenesis, steps of angiogenesis that will include angiogenesis from pre-existing blood vessel as well as from endothelial precursor cells, and we will also talk briefly about the different roles of growth factor in angiogenesis and finish our discussion with the dreaded topic of Notch signaling pathway in angiogenesis. Okay, so a lot of topics, so let's begin. First question, what is angiogenesis? Now the word angiogenesis has two parts. Angio means blood vessels and genesis means formation. So from the meaning it is very obvious. How can we define angiogenesis? Angiogenesis can be defined as the process of new blood vessel development from existing blood vessels. So I am repeating the definition again for my students. What do we mean by angiogenesis? Angio means blood vessel, genesis means formation. Angiogenesis can be defined as the process of new blood vessel development from existing blood vessels. Now notice the term existing blood vessel, because although we are saying that in the definition, but as we will see later, angiogenesis can also happen sometimes from endothelial precursor cells as well. So now that we have defined angiogenesis, now we will move on to the next topic and discuss some terms. The first thing that you have to know is angiogenesis is also sometimes called neovascularization. Neo means new, and I'm sure you know what vascularization means. So they are similar in meaning, angiogenesis and neovascularization. Both means formation of new blood vessel or development of new blood vessels from existing blood vessels. Now don't confuse the term angiogenesis with vasculogenesis. We'll also see the term vasculogenesis in your textbook, and always remember the term vasculogenesis was used to denote blood vessel assembly during the embryonic development. And always remember, during the embryonic development, initially we have primitive vascular network. Those primitive vascular networks were established from different types of precursor cells. And always remember, whenever you see the suffix blast at the end of the name of a cell, you have to remember that that thing is a precursor cell, or that thing can be a stem cell. So coming back to today's discussion, what do we mean by vasculogenesis? It denotes assembly of blood vessels during embryonic development. And during embryonic development, we will see some precursor cells, say for example angioblasts or hemangioblasts, and from there primitive blood vessel will derive, and that process is known as vasculogenesis. Now before moving on to the next topic, I want to focus your attention on this interesting cell, hemangioblast. Now can you tell me what type of cell can we get from this primitive stem cell? Should we get blood cells like red blood cell, white blood cell and platelet, or should we get something else? Yes, look at the term hemangioblast. We have both hem and angio in this word, so it is a dual precursor cell. It will give rise to hemopoietic cells that will later give rise to different cells of the blood, cells like red blood cell, white blood cell, platelets, etc. But at the same time, hemangioblast will also give rise to endothelial precursor cells, and that will give rise to primitive blood vessels. So that was a nice thing to know. So now that we have defined angiogenesis and also briefly talked about some common terms, now we will move on to the next topic and discuss the roles of angiogenesis. Why do we need angiogenesis? Now, angiogenesis has both beneficial and harmful roles in our body, as we will see. For example, during the healing process, new blood vessels must be developed, and that thing is achieved by angiogenesis. So that is a beneficial effect of angiogenesis. Similarly, angiogenesis plays a vital beneficial role during the development of collateral circulation during ischemia. Now, what is ischemia? Ischemia means reduced blood flow to the tissue, and whenever there is reduced blood flow to the tissue, we need to develop additional blood vessels to supply that affected area, and that thing is achieved by angiogenesis. So these were the beneficial effects of angiogenesis. Always remember about the role of angiogenesis in wound healing as well as in collateral circulation. However, angiogenesis also has some harmful effect as well. For example, in neoplasia or tumor, sometimes the neoplastic or tumor cells can also induce angiogenesis, and that will help the tumor to grow. And although that is helpful for the tumor, but that will be harmful for the host. Now, I have two separate videos on neoplasia or tumor, and you can also look into those two videos after watching this video for more information. And I also have two videos on wound healing that you can also watch after finishing this video for more information. So another harmful effect of angiogenesis is sometimes it can cause a lot of disease. For example, angiogenesis, pathologic angiogenesis in the blood vessels of the retina can result in retinopathy, and this is particularly seen in diabetic retinopathy. So these were the harmful effects of angiogenesis. So now that we have discussed the different roles of angiogenesis, now we will move on to the next topic and discuss the steps of angiogenesis. So first we will talk about the different steps of angiogenesis from pre-existing blood vessels. So here I am showing you different images depicting different steps of angiogenesis. On the topmost figure you can see a blood vessel, a pre-existing blood vessel or a quiescent blood vessel. New blood vessel will develop from this blood vessel. Now notice what has happened in the second image. You can see that the same quiescent blood vessel has now dilated, and there is also increased vascular permeability. So these were the initial steps of angiogenesis: vasodilation and increased vascular permeability of the quiescent blood vessel. Now, why will there be vasodilation? That will be due to nitric oxide. Always remember, nitric oxide is a potent vasodilator. And at the same time, there will be increased vascular permeability, that is mediated or induced by VEGF, that stands for vascular endothelial growth factor. So that was the first step of angiogenesis. Now the next step in angiogenesis is separation of pericytes. Now can you see the pericytes on the image? Notice in every blood vessel from inside to outwards we have endothelium, then there is basement membrane, and outside that basement membrane we have some periendothelial cells, and some of them are called pericytes. And always remember that in larger blood vessel there can be also smooth muscle cells present around the endothelial cells as well. So coming back to today's discussion, the next step of angiogenesis was separation of pericyte from the abluminal surface, and also there will be breakdown of the basement membrane. And who will break down the basement membrane? Matrix metalloproteinase, or MMPs, which are enzymes present in the extracellular matrix. They will help in the breakdown or degradation of the basement membrane. The next step in angiogenesis will be migration of the endothelial cell toward the tissue injury. So here you can see in the next image we are seeing migration of endothelial cell. They are migrating towards the stimuli, and what is happening behind them? Behind the leading front, or behind the tip of the migrating cells, we can see proliferation of endothelial cell. So it's like a leader is leading and the remaining endothelial cells are following him. Wherever the leader will go, they will go. Wherever the leading front will go, the proliferating endothelial cells will follow. The next step in angiogenesis will be remodeling of these migrating cells into capillary tubes. And initially the tube will be leaky. So how will we stabilize the tube? There will be recruitment of periendothelial cells. Just like I had mentioned previously, there will be recruitment of pericyte for small capillary and smooth muscle cells for large vessel, and they will form mature blood vessel. And the last step of angiogenesis will be suppression of the endothelial cell proliferation and also suppression of migration of the endothelial cell, and there will be again deposition of basement membrane that was previously degraded by matrix metalloproteinase. So this was in short about the different steps of angiogenesis from pre-existing blood vessels. Angiogenesis can also happen from endothelial precursor cells, or EPC. So the endothelial precursor cells are present in the bone marrow. So first they have to be recruited from the bone marrow into the site of injury, or into the site where we need angiogenesis. And the exact mechanism of these homing phenomena is still uncertain. Just remember that endothelial precursor cells influence the vascular function, and they also determine the risk of cardiovascular diseases as well. And also remember that this type of angiogenesis has role in neovascularization of ischemic tissue as well as in tumor and wound healing. So now that we have talked about the different steps of angiogenesis, now we will move on and talk briefly about the roles of growth factor in angiogenesis. So the major growth factors involved in angiogenesis will include vascular endothelial growth factor, especially vascular endothelial growth factor A, fibroblast growth factor, mainly fibroblast growth factor 2. There will be also role of angiopoietin 1, angiopoietin 2, and also platelet-derived growth factor and TGF-beta in angiogenesis as well. So let's talk about their function one by one in angiogenesis. So what is the role of vascular endothelial growth factor A? This growth factor stimulates migration and proliferation of endothelial cell. So that will help in angiogenesis. At the same time, it promotes vasodilation by indirectly stimulating production of nitric oxide. And we all know that nitric oxide is a potent vasodilator, and vascular endothelial growth factor will stimulate production of nitric oxide and help in vasodilation as well. And that will also contribute to formation of the vascular lumen. Fibroblast growth factor 2 stimulates proliferation of endothelial cell, and one additional function is it also promotes macrophage and fibroblast migration to the affected area. Moving on to the function of angiopoietin 1 and 2, always remember that they help in the structural maturation of the newly formed blood vessel. Like I said previously, the newly formed blood vessels are leaky and they need to be stabilized, and by stabilization we mean recruitment of smooth muscle for larger blood vessel and recruitment for pericyte for smaller capillaries and similar blood vessels. So this is stabilization of the blood vessel, and angiopoietins 1 and 2 will have role in that. Regarding the function of platelet-derived growth factor, that will recruit smooth muscle cell, and TGF-beta will suppress endothelial proliferation, migration, and that will also enhance the production of extracellular matrix protein. So these were in short about the roles of growth factor in angiogenesis. The last topic that we will talk about today is regarding the notch signaling pathway. Now, a lot of my students are afraid of this signaling pathway and they tend to run away when I try to teach this topic. I even have to show them teddy bears to keep them calm. So look, I'm also showing you a teddy bear to keep you calm. So look at the teddy. Don't get scared, because we will try to discuss about this thing briefly and in a simple manner. So what is the role of notch signaling pathway in angiogenesis? Now, in order to answer that, first you have to ask a question. Recall from my previous discussion about the steps of angiogenesis that during angiogenesis a lot of things are happening. Still, the newly formed blood vessels are able to maintain a perfect pattern of blood vessel, and this thing is achieved by the notch pathway. The function of notch pathway is it promotes the proper branching and sprouting of new blood vessels, and at the same time it prevents excessive angiogenesis. How does it prevent excessive angiogenesis? By decreasing the responsiveness of vascular endothelial growth factor on the Stalk cells. Now, what is a Stalk cell, you may ask? Now, in order to answer that question, now I will show you the image again where we had seen angiogenesis. So recall, in the image we had two types of cells. The leading cell was called the tip cell. That cell is the leader. That cell was proliferating, migrating towards the site of injury or towards the site where the new blood vessel must grow. At the same time, there was another type of cell which were called Stalk cells. And Stalk cells, their function was different. Stalk cells maintained their connection with the pre-existing blood vessel. So we can see two different patterns here. You can see that the tip cell is migrating and proliferating to go to distant site, and the Stalk cells, they are maintaining the connection. Now, in order to do that, the Stalk cells must have a dampened response to vascular endothelial growth factor, because if the Stalk cells also begin to migrate like the tip cell, the perfect pattern of the blood vessel will be lost. So that is the mechanism that we will see here in the notch signal pathway. So in the notch signal pathway, we will have notch ligands and receptors. In mammals, there are 5 notch ligands. They are called delta-like ligand or DLL1, 3, 4, and also there is 2 other ligands called the JAGD1 and JAGD2. Now don't get scared, you just remember these 5 names and nothing else is required for your exam. So just remember that in the notch pathway there are ligands and receptor on the cell surface and they interact. There are 5 notch ligands. They are the delta-like ligand 1, 3, 4 and JAGD1, JAGD2. And there are 4 notch receptors. They are transmembrane receptor. They have portions that are extracellular and also they have intracellular portions, and they are named notch receptor 1, 2, 3 and 4. So now I am going to show you an image of notch signal pathway, and you can see that we have different things happening here. Shown on the upper side, we can see the tip cell, and on the cell membrane of the tip cell we can see there is different types of receptor. One of the receptor is vascular endothelial growth factor receptor 2. Now vascular endothelial growth factor will bind with this receptor, and that thing will result in signal transduction, that will result in gene transcription, and ultimately there will be upregulation of delta-like ligand 4 mRNA. So I am repeating again because this thing is a bit difficult. So on the top side we can see the cell that will be the tip cell that will involve in migration and proliferation of angiogenesis. So we can see first the growth factor is binding with the receptor on the surface of the tip cell, that is causing signal transduction, gene transcription, and as a result of gene transcription we can see there is increased number of delta-like ligand 4 mRNA, and that will make the protein that will form the transmembrane delta-like ligand 4. Now on the bottom side you can see the stalk cell. The stalk cell has notch receptor, and look what is happening when the notch receptor of the stalk cell is interacting with the delta-like ligand of the tip cell. That thing will result in several phenomena. Binding of the notch receptor with the notch ligand will result in proteolytic cleavage of the notch intracellular signaling domain. That was the intracellular portion of the notch receptor. So that thing will now undergo proteolytic cleavage. It will move in the cytoplasm and then enter the nucleus, and there it will cause gene activation. The activated genes will dampen the responsiveness of the stalk cell to vascular endothelial growth factor. So this is in short about the notch signaling pathway. Through cross-talking with the vascular endothelial growth factor, this pathway will control the sprouting and branching of the new blood vessel and also prevent excessive angiogenesis. And the main idea of these notch signaling pathway is to ensure that whenever new blood vessels are formed, they are formed with proper spacing in between them, and that helps in effective blood supply to the healing tissue or to the injured tissue. So this concludes today's video on angiogenesis. I hope this video was helpful. If you like my videos, do comment, share, subscribe and let me know. And for my students, I will always recommend you to go through your textbooks to know more information. So that's all for today. Until next time, take care and stay blessed. Thank you.